testing station characterizing photosensors for the pfRICH for the EPIC collaboration at EIC. So the EIC, or the Electron-Ion Collider, is a collider that will be built or scheduled to be built at Brookhaven National Lab in 2035. And the EPIC detector is a detector that will be built within the EIC. And within the EIC, as you can see in this picture here, it contains a lot of sub-detectors. And for the purpose of my research, I will primarily focus on the proximity-focused rich, or pfRICH sub-detector. This sub-detector is primarily used for particle identification, or PID, and it is used to identify particles like protons, electrons, muons, pions, and so on. And so within electron and ion collisions, there'll be some sort of scattering, so collision products. And some of these will be in the electron going direction, which is indicated by the electrons arrow going this way. And these particles will then be picked up by the pfRICH. And then it is the pfRICH's job to identify what these particles are. So how does the pfRICH actually identify particles? It does this through something called Cherenkov radiation. And what this means is that when charged particles, or these scattered particles, go into the pfRICH, they'll enter a layer of this radiator, which is chosen to be aerogel. Aerogel has a refractive index of around 1.045, which is extremely close to air. And it is chosen so that when these charged particles go into the radiator, they're faster than the speed of light in aerogel. And when this happens, they will emit Cherenkov radiation, or Cherenkov photons, which are just photons emitted in a cone shape around the charged particle's path. This is kind of similar to the supersonic boom produced by jets flying faster than the speed of sound. And so when these photons or cones expand throughout this expansion volume, they will eventually be detected by the photosensor plane and detected as signal. So depending on the different particles and their corresponding momentum, the sizes of these cones will change. And one thing that changes is the Cherenkov angle, which is the angle between the charged particle's trajectory and the corresponding photons that it emits. And so if we are able to correctly measure this Cherenkov angle, then we can calculate the velocity of the charged particle. Additionally, the momentum of the charged particle is also calculated by a specific tracking system that is not particular to the pfRICH. But if we graph that momentum with the Cherenkov angle, then we could produce a graph like this, which is really crucial for particle identification, because each one of these curves here represents a specific particle. And so how do we ensure that the pfRICH is actually doing its job? Well, it needs to have a large expansion volume, which is the volume between the radiator and the photosensor wall. So when we have a large expansion volume, then these cones spread out more, and it is easier to differentiate between cone from cone. And we also want a thin radiator, because then it is easier for us to trace back exactly where the Cherenkov radiation was emitted from. We want a high spatial resolution in the photosensor matrix. That is self-explanatory. And lastly, we want there to be enough photons in the first place to be measured. So the specific photosensor that we use is called the HRPPD, and it is manufactured and designed by this company called Incom. And they specifically developed the HRPPD for the pfRICH sub-detector. So this is a relatively new technology, and it has two main features to it. We are able to measure its absolute quantum efficiency, as well as its gain map. I'll speak more about what those are in the next slide. So how do we actually test this HRPPD? Because it's such a new detector, there has to be some sort of way to characterize these photosensors. So instead of reproducing the Cherenkov radiation, which is extremely expensive, we are instead using LEDs and focusing these LEDs onto a beam splitter. And some portion of this beam will then be reflected to a photodiode and the rest into an HRPPD. So when these photons arrive at the HRPPD window, which is at the very top layer of the HRPPD, they emit electrons into the HRPPD. And then these electrons are then what goes into the anode ground plane and then detected as signal. So if we compare the current that we measure from the photodiode with the current that we measure from the HRPPD, that is exactly how we measure the absolute quantum efficiency of the HRPPD. Another way to put this is if we send 100 photons into the HRPPD and we measure 30 of those photons, then it is said the HRPPD has a quantum efficiency of 30%. Something else we can do is we can attach high voltage cables into the HRPPD, specifically towards the microchannel plates with the MCPs, where they create such a strong electric field. So when the electrons go into this MCPs, they begin to multiply, creating something called an electron avalanche. And the purpose of this electron avalanche is so that fainter signals get detected easier. So naturally, one thing that we would want to measure is by how much these electrons are getting multiplied by. And this is exactly what I meant when I said we wanted to measure the gain map of the HRPPD. So how much of the setup do we currently have right now? We have a DAQ computer, which is used to store and collect all of the data that we collect from the experiment, as well as automate the translation stage. I'll talk about more about the translation stage on the next slide. We have a NIM crate. The NIM crate essentially sends pulses into the LED to pulse the LED and sends an identical pulse to the DAQ computer to let it know when the LED is pulsing, so it begins collecting data. And we have an oscilloscope, which allows us to look into the experiment at any particular time to make sure that everything is running smoothly. We have a VME crate, which houses the digitizer. The digitizer is what converts all of the analog data that has been collected by the HRPPD and converts that into digital data that is readable by our DAQ computer. We, of course, have a smaller version of their dark box. And the purpose of this dark box is so that we're able to isolate our experiment because after all, we are detecting photons. So we don't want the lights in the room to have an effect on our data collection. We have a high voltage power supply because as I was saying earlier, if we wanted to measure the gain map of the HRPPD, we would need to supply it with high voltage. And this is exactly where it comes from. We also have a translation stage, which consists of 3 different motors. And we stack them on top of each other in such a way that we have all X, Y, and Z directional movements. And one thing I left out about the HRPPD is that it consists of 32 by 32 pixels, and we are aiming our LED at 1 pixel at a time. So the purpose of this translation stage is to go through all 1,024 pixels, and in the end, tell us about the quantum efficiency or the gain. But of course, it's not as easy as just shining the LED onto the HRPPD. As I was saying, we needed to focus the LED, so we need a focusing lens. We need a beam splitter. We need a photodiode, which is why we have an entire optics setup dedicated to this, as you can see in this CAD right here. And these 2 pictures up top describe our optics setup in more detail. Essentially, what happens is we connect an optical fiber to our pulsed LED and use the other end to connect to a lens tube. So, which can be seen as the blue LED in that picture over there. And of course, the light rays are going in all sorts of directions, which is why we need a convex lens there to collimate the light, which is to put all the lights in parallel with each other. And then this collimated light is what goes to the beam splitter, put some of it to the photodiode and the others into the HRPPD. This is how much of the setup we currently have right now. We have the lens tube, which houses the lens. We have a cage cube, which will eventually hold both the beam splitter as well as the photodiode. But unfortunately, both the photodiode and the beam splitter are being shipped as of now. This is just a quick demonstration showing that the focusing lens does in fact collimate the light. As you can see here, there's a very small dot. And what are some of the things that we would like to change or alter about our optical setup? Well, right now we only have 1 LED that is of 4 or 5 nanometers. But of course, not all of the Cherenkov radiation will be emitted at the same wavelength. So ideally, we would want multiple LEDs of different wavelengths, around 200 nanometers to 600 nanometers. And with that, we would also need a better photodiode to compensate for this wavelength range, which is this blue cabled one right here. And something else we would like to add is this reflective collimator, which is best described by that picture over there. So after the beam splitter, this collimated light will go into this reflective collimator, and then it gets reflected downwards into a focal point. And at this focal point, we connect an optical fiber to it and use the other end to shine the light out of it. The purpose of this, or rather the advantage of this, is so that our entire optical setup is stationary within the dark box, and the only thing that is moving or connected to the translation stage will be this output optical fiber, which then is used to scan all 1,024 tiles of the HRP or pixels of the HRPBD. Unfortunately, we also don't have the HRPPD income. Its company, the manufacturer, has said they will ship it to us this summer. Unfortunately, like it was looking more and more ambitious every single day. But we still have much more to do on our optical setup. We have to calibrate our photodiode, ensure that all of the optics are aligned, as well as implement this reflective collimator into our setup. So just a quick summary. I've been working on this testing station since last semester, where I was primarily doing the automation for the translation stages, testing high voltage cables, and beginning to work on this optical setup. But this summer, I finally finalized the optical setup, which took a long time. Began putting together the parts, which are coming in slowly. And also something else that we'll be working on is setting up the digitizer, making sure that it runs with the NIM crate and our AAQ computer. And hopefully by next semester, the HRPVD will be here and we are able to take some measurements. And something to look forward to is getting different wavelength LEDs so we can take these measurements at different wavelengths. And just before I conclude, I'd like to bring your attention briefly to a side project that I've been working on, this Van de Graaff generator. If you wanted to know more about it, I'm open to speak about it more after this. It seems like I'm running out of time, so.
Any questions? Thank you.